Hello, today we're going to be doing Digital NCBI Database Laboratory. The goal of this lab is to improve your understanding of relationship between genes, proteins, and biological functions. So in today's lab, you will learn to retrieve information about the gene, DNA, mRNA and protein from genome databases at the National Center for Biotechnology Information, NCBI. So let's start. The first thing you do is start your internet browser and go to www.pubmed.com. So PubMed is a part of the NCBI database. PubMed is a library of journals that contain primary source literature, primary source articles about biomedical discoveries about genes and proteins and health-related articles, as well as other scientific articles. So it has a lot of peer-reviewed literature and just science literature. So in the future, when you write your final paper or prepare your PowerPoint presentation and you need to find a primary source literature for the topic that you're presenting, you should go to PubMed and then you would type your topic here, or your gene or your protein name, and press search, and then you would get a list of all the articles published about this topic. For example, if you're interested to learn about papers on gene therapy, you would click on here, and here you would get various papers about gene therapy. So, but in our lab today, we're not going to be using PubMed. Instead, we're going to be learning about your gene. So all of you got assigned a certain gene, and so let's do the first step in your lab, which is so the first step is to go to gene. So click on the scroll down menu and select gene, and then type your gene name. So I will be using GTF2I gene as an example. So click search, and here you have GTF2I, which is a gene in humans, and you can see that this gene is also present in mice. So musculus stands for mouse in Latin. This is Ratus norvegis. This is a Latin for a rat, and so on. And it has all kinds of animals here that have this gene. So we're interested in human version of this gene. So let's press on this link. And now for your lab report, please in your first step copy the code name of the gene. In my case it's GTF2I, and then the full name for the gene. In my case it's general transcription factor 2I. And then the next thing you want to copy is other acronyms or other names for this gene, because different research groups may have discovered this gene at different times, and its sequence may have been slightly different depending on how they clone the gene. And so here's some alternative names for this GTF2I gene. So it's also known as WBS and TF2I and BAP and so on. So in my report, I will also include this line here, which contains the alternative names for this gene. So the next step is step 2, and it asks you to write a short essay which would describe the function of your gene and its involvement in human health and human disease. So how do you find out that information? So go back to the scroll down menu, and we're gonna select OMIM or MIM database. So OMIM, and put in your gene name again, GTF2I, and click search. So now you see that there's a lot of information available about this gene, and you can click on any of these links and they might have different information from different journal articles which are collected in OMIM. So OMIM is Online Mendelian Inheritance in Man, and it basically tells you about how a certain gene or a protein are involved in health and disease, in human health and disease. So let's press on this link, for example. So here we have our gene name again, and it talks a little bit about the discovery and the cloning of this gene. As you remember, cloning a gene just means putting a gene into a plasmid and then learning about its function and so on. So here it says that the gene was discovered actually by my graduate school mentor, Dr. Roy, and then it talks about gene function, so it's a transcription factor, and then this gene is actually involved in something called the Williams-Beuren syndrome, which is a very interesting syndrome. It's a neurodevelopmental disorder. Unfortunately, it involves sometimes mental retardation. One interesting thing is people with this syndrome have difficulty doing math problems or recognizing shapes, but their language development remains intact. In fact, they sometimes have higher than expected for their age vocabulary, and they're very friendly, overly friendly in social interactions. What that means is sometimes they're so friendly that they might not understand such concept, for example, as not talking to strangers. So people with this syndrome have some aspect of autistic personality as well. So it's a little bit related to the autistic spectrum personality. So this, as you can see here, it says that this syndrome results from a deletion of a chunk on chromosome 7, and this chunk of this chromosome contains a gene for TF2I. So the gene for TF2I is deleted in Williams-Beuren syndrome, and so on. And here it talks a little bit about the molecular mechanism of TF2I. So in your report, you should talk about the function of your gene. So what kind of protein is it? Is it a transcription factor that can bind DNA, or maybe it's a tumor suppressor protein that participates in the cell cycle and DNA? Maybe it's an oncogene and it actually speeds up the cell cycle. Is it a maybe it's a DNA repair protein? So you should discuss the function of the protein. And then the next thing you should do is talk about the molecular pathway that this protein is involved in. For example, if the protein is involved in bone development, as TF2I, for example, TF2I is involved in bone development, you should talk about that. And then the third thing, you should also mention if a mutation in your gene may cause or may predispose people to certain diseases, or if it's important in health, such as it might be important in aging or obesity, perhaps, something like that. So the next part of your report is step 3, and so let's go back to gene here. So gene, and press go. So let's click on this gene again. And so the next thing we want to do is view this gene on a chromosome. So click on Map Viewer. And here it shows you the position of TF2I gene on chromosome 7. So copy this into your report, 7q11.23. So what in your case this would be a different number here, and it might have either Q or a P letter here. So the first number here, 7, this stands for the chromosome number. So it could be, you know, we have 23 chromosomes, so it could be we have 22 autosomal chromosomes and one pair of sex chromosomes. So it could be a number from 1 to 22, or it could be X or a Y. So in my case it's chromosome 7, and Q stands for the long arm of chromosome 7. If you have a P here, it means the short arm of your chromosome. And here's the region where the gene is located. So what we need to do for the lab report is copy this picture of the chromosome. It's showing you the chromosome itself and these bands, which are various bands on the chromosome, and your gene is located here. So these bands are generated because of a special stain that can be used to color chromosomes, such as Giemsa stain, for example. So here's my gene. It's located right here. And so what I will do for the report is get an image of this. I will use the snipping tool and just select this and save it. Okay. And so in your report you'll have this picture, and under the picture you will put the caption, which is, in my case it's gonna be 7q11.23, and then the full name. Chromosome, my gene is located on chromosome 7, long arm, region 11.23. So this is your step 3 of the lab report, and now let's move on to step 4. So to do that, let's go back to the gene again. So here it says gene, and this is my gene here. And so the next thing is we want to look at the sequence of the gene. So we're going to press here on gene bank, and if you scroll all the way down here, you will see the gene sequence, and you see how long this sequence is. In my case it's about 102,000 base pairs, so it's very, very long. I don't need to copy all of that into my lab report. So what I need you to do for the lab report for step 4 is to copy the full name of the gene, which in my case is transcription factor 2I, general transcription factor 2I, and then put the NCBI database ID number for the gene, which is NC00007 in my case, and you will have a different ID number. It will start with NC and then it will have a number, and then insert the base pairs. So this is 102 and 993 base pairs DNA, and so you will have the gene name, ID number, the size of the gene, the length of the gene at base pairs, and then a link, this link. So this link will take me to the sequence of your gene. Why is this gene so long? It's because it not only contains exons, which are coding sequences, it also contains introns, which, as you remember, are non-coding intervening DNA sequences, which are removed later when mRNA is transcribed, when mRNA is processed. So we're done now with step 4, and let's go to step 5. So for step 5, we talked about the gene now and about the DNA, which contains both, as I said, exons and introns, the coding and the non-coding sequences, and that's why it's so large. But now we're going to talk about the mRNA. So let me take you back for a second. So if you look at this, so that we're back on the gene page. This is our gene name, and as you remember, there were these 2 diagrams here. So this diagram is just showing you again the location of the gene on the chromosome 7, but in this case it's showing you kind of a zoomed in view, and it's showing you the TF2I gene and other neighboring genes which are close to the TF2I gene. So you don't need to copy this into your lab report, but I'm just showing it to you, because it's interesting. And here you have another diagram, and this is showing you that TF2I gene actually encodes 5 different RNA isoforms. So if you, during the process of splicing, introns are spliced out and exons are joined together. So you can see that you can shuffle the exons in different ways and produce 5 different isoforms, 5 different versions of mRNA. And this accounts for 5 different versions of the protein. So NM here stands for the mRNA, and that corresponds to these proteins here. So as you can see, splicing accounts for the diversity that we see in the sense of proteins. So we have only about 20,000 genes in our genome, but we have thousands and thousands of proteins, and this is because one gene can produce different variants, different isoforms of proteins and mRNA. So first you have 5 different mRNA isoforms, and then they're going to be translated to make 5 different protein isoforms of this gene. So in any case, let's go back to GenBank here. So press on this link, GenBank. And again, you didn't need that diagram with the green arrows. I was just showing it to you to explain that there can be different isoforms of the same gene. So we're back on the GenBank page of for your gene, and so let's scroll down, and now we are on step 5, and we're going to be looking at the mature mRNA. So here, let's look at the first mRNA that you encounter when you scroll down, and you're gonna press on this link, which goes just right after, which corresponds to this mRNA. So there's this NM link here. So let's click on this, and this will take us to the page that talks about the mRNA. So for your lab report, copy the name of your mRNA, which is in my case called General Transcription Factor 2I Transcript Variant 1, and it's an mRNA, and then copy the NCBI code that corresponds to this mRNA, and also the base pairs. There's 4,529 base pairs in this mRNA. And here you should also mention, you should compare this size of the mRNA to the size of your gene and comment on whether mRNA is significantly smaller or not than the actual gene. So in my case, the gene was 102,000 base pairs, and you can see that the mRNA is a lot smaller. It is a lot shorter in length than the gene. So then the next thing you want to do is actually look at the graphical representation of your mRNA. So we're gonna click on graphics here. So here you will see the mRNA, and it's showing you all the exons that are joined together to make this variant 1 mRNA. And what we need for the report is just this top part here that's showing all the exons. If you look below here, what this is showing you is the whole TF2I gene, and then this is the protein, and it's showing you different sites on the protein that can be modified by addition of various residues that can be phosphorylated, or there's also something called a nuclear localization signal that allows the protein to go to the nucleus, and this is a very important part for transcription factor, because in order to help transcribe and activate genes, it has to be able to enter the nucleus and has to be directed to the nucleus. So this nuclear localization signal domain of the protein directs it to the nucleus, and phosphorylation usually helps to activate the protein. make it active. So we're not gonna worry about this part, but what we need to do is again get the snipping tool, and we're gonna get this part for our lab report and save this as a file. And so you will save this and include this into your lab report, and underneath you will write a caption saying that this is a graphic representation of your mRNA, and then you will say it contains the following exons, and you just list the number of the exons. So exon 1, so you'll just say it contains the following exons, colon, and then say exon, and then just list the numbers 1, 2, 3, 4, 5, and you'll notice that some of the numbers are missing because those exons were not included in this mRNA isoform. So mRNA isoform 1 might not have, suppose, exon 15, but variant 2 might have exon 15. So we're not gonna keep this. I'm just showing you the file that you would need to generate. So now at this point we're done with step 5, and we can go to step 6. So just press the back button here, and we're gonna press back again, and now we're back at on the gene bank gene page that had the NC, remember, ID number. So if you're lost, you can also just go to gene and then type your gene name. So again, we're back. Here's our gene, and then we'll press on gene bank, and here we are. So this is the page that we need. Now let's scroll down and find our mRNA again. So here's our mRNA, and you see how here it says join. So what this is listing is the exons and the position of each exon in the mRNA. So exon 1 is located between 1 and 365. Exon 2 is located between 31429 base pair and 31,532,000 base pair. So what you need to do for your lab report is copy this piece of information, and you'll save it into your lab report. And so let's open this. So suppose I saved it, and this should really go kind of above this diagram here. But yeah. So the first piece that you need to answer step 6 is this piece of information. And now we're gonna make a diagram showing the positions of exons and introns in our mRNA. Now if your mRNA has more than 10 exons, which it looks like my mRNA certainly has more than 10 exons, you only need to do the first 10. So how do you draw this diagram? So you can do it in PowerPoint, or you can do it by hand. So here you have exon 1, and the position of this exon in mRNA is from the first nucleotide 1 to 365 here, and intron 2 is between 31429 base pairs and 31,532 second base pair. So here you have exon 2, and the intron you can draw between these two boxes. So the intron is kind of the comma here, so the in between of these two regions. And then exon 3 would be from 33276 to 33414. So hopefully you get this point here. So the diagram just shows you the location, the position of exons and introns. But now we need another thing that we want to do for the lab report is to calculate the size of each exon and intron. So how do you calculate the size of exon 1? So that's between 365 and 1, but the nucleotide 365 is still included as a part of your exon. So that's why it's 365 minus 1 plus 1. So it's 365 base pairs. So the general formula is the ending base pair minus the starting base pair plus 1. And it's all in base pairs. And then the exon 2, it would follow that to calculate that you subtract again the ending base pair minus the starting base pair, 31429 plus 1. And to calculate the size of the intron, you subtract kind of this first base pair of the following exon minus the last base pair of the previous exon. So in this case it's 31429 minus 365, and you don't add 1 in this case because 31429 is already a part of the exon 2. So what you need to do in your lab report is first of all have this excerpt from the database, then have the actual diagram of the positions of exons and introns, and finally calculate the sizes of each exon and each of the introns. So you will have 10 exons and 10 introns, and you can show how you calculate the first 2 exons and the first intron, so that I know that you understand how to calculate the sizes. And then you don't have to show me the calculations for all of them, just give me the numbers. Okay, so we're done with step 6, and in the next lecture I'll talk about step 7 and 8 and 9 and the rest of the lab and step 10. So okay, bye.